Our minds are what our brains do. So what do our brains do? We don't know. But that doesn't mean we know nothing. One thing we're certain about is that the most basic unit in whatever it is the brain is doing is the nerve cell or neuron. It's still a mystery exactly how these work together to create perception, thought, learning, complex actions, feelings, ideas, and other parts of our mental experience. But knowing how one neuron works could be an essential key to unlocking the rest of the mysteries of the mind. Greetings, intelligent creatures. Welcome to Behavioral Logic, where we explore intelligent behavior in theory, practice, computers, and neuroscience. Before you click away, if I could just have 1 minute of your time, I will give you the too long didn't watch version of how neurons work. Neurons are nerve cells in your body and other animals that, 1, receive information in the form of chemical signals from other neurons, or, in the case of sensory neurons, from external conditions like light, temperature, movement, or chemicals in its environment. 2, perform some kind of calculation with that information. 3, based on the results of the calculation, either it sends or does not send an electrical signal down the length of its body. 4, optionally, it can make persistent changes to itself in how it receives, calculates, or transmits information. And 5, it can secrete a chemical signal to other neurons, or, in the case of motor neurons, to a muscle, gland, or other organ. If that's all you want to know, well, you're free to go. Personally, I'd like to know a little more information, so I'm going to spend the rest of this video diving deeper into each of those steps. There's no way I'm going to be able to talk about everything that we know about neurons, but I will do my very best to present the most scientifically rigorous information needed to help form a decent understanding of the basic neuron's function. If that sounds interesting to you, then please join me, or, at least leave me on in the background while you go do something else. That's fine, I don't mind. Let's start with step 3, because I think that is the simplest one to explain. The long, thin part of a neuron is called the axon. How long is it? Well, in your body, the sciatic nerve is a bundle of axons running from the base of your spine to your toes that can be over 1 meter long. That's about a yard for us folks in the United States. Blue whale neurons can be over 30 meters long. Impressive, especially because individual axons are typically only a few millionths of a meter thick. An electrical signal that travels down the length of a neuron is called axonal propagation. It's a relatively simple process and fairly well understood compared to a lot of the other processes and functions of the neuron. So I'm going to take the opportunity to really dig in and understand what's happening at the molecular level, which means first, we're going to have to talk about molecules. Yes, molecules. I said I wanted to take a deep dive, and I wasn't lying. If I went any deeper, we'd be in physics. Here's what I think that we should know about molecules for us to really understand the mechanisms of axonal propagation. Molecules are tiny structures made from atoms that are stuck together. Here is one molecule called N-hexane. It has 6 carbon atoms and 14 hydrogen atoms bonded together in this arrangement that I've represented using letters and little sticks. It has the chemical formula C6H14, but that doesn't tell you anything about the specific shape of how those atoms are connected, only which atoms are in it. It's also common to see organic molecules drawn with a zigzag line like this, where each end and corner represents a carbon atom, and if it is not explicitly shown to be bound to at least 4 other things, then we're going to assume that there are hydrogens making up the difference. But this isn't really how a molecule looks. That's impossible. You can't see atoms. They are smaller than the wavelengths of light we use to see anything. But we can infer the structure and draw models that sort of make sense to our brains. N-hexane is probably shaped something more like this. Now let's take a look at the structure of a few more common molecules. Water, H2O, is made from sticking 2 hydrogen and 1 oxygen atom together. Notice how the molecule is a little lopsided? This makes it polarized. It has a more negative side over by the oxygen and a slightly less negative side over by the hydrogens. Comparing this to N-hexane, where every spot on the molecule looks pretty much exactly like every other. Because water has 2 differently charged sides, it can be described as polar, and hexane as nonpolar. Table salt is sodium chloride, made from 1 sodium and 1 chlorine atom. Sort of. Not really. I mean, salt is made from atoms with electrical charges called ions. Sodium has a +1 charge. It is a positive ion, or cation, and the chlorine has got a -1 charge. In its anionic form, we call it chloride. Because they have opposite charges, these atoms stick together pretty well, but they're just as happy to stick to anything else with an opposite charge. This molecule is so polar that its differently charged parts can just sort of wander off. Let's talk about another molecule now. Here's the zigzag version of oleic acid, the most common molecule in canola oil. It's basically like a long hexane with some oxygens on one end and a double carbon bond in the middle. It's considered to be nonpolar, but can we prove it? Let's test it. We know that polar substances will happily mix together, and nonpolar will also play nice with themselves, but they don't mix with each other. But why? Polar molecules and ions are attracted to their opposite charge. We can also refer to polar substances as hydrophilic or water-loving. And that includes water. It loves itself. Isn't that nice? Oleic acid is nearly all carbons and hydrogens, so it's considered nonpolar. Yeah, it's got some oxygens on one end, but I guess that doesn't make it polar enough. Nonpolar molecules, like oils, are described as hydrophobic or water-fearing.
Hydrophobic.
Polar substances will happily mix together. And nonpolar substances, well, they'll also play nice with each other. But they don't like to mix with each other.
Oh, no, Travis.
Now let's take a look at the structure of a molecule called sodium lauryl sulfate, also known as SLS or sodium dodecyl sulfate. NaC12H25SO4. Wow, these molecules are getting kind of big. It is the most common ingredient in dish soap. One side is this very long nonpolar carbon chain, but the other side has got this sodium atom and a bunch of oxygens. It looks a lot more like the structure of water and salt. SLS is referred to as an amphipathic or amphiphilic molecule. It likes both polar and nonpolar substances. And we all know what happens when you add soap to a mixture of oil and water. Why am I telling you all of this? Because amphipathic molecules like soap are what the cell membrane of neurons and other animal cells are made of. Now, we are ready to start talking about neuronal membranes. We already know that polar and nonpolar molecules only like to hang out with themselves. But how do amphiphilic molecules feel about each other? That's an interesting question. See, when they're next to each other, molecules like SLS will arrange themselves so that the polar and nonpolar ends gather together with each other. This will cause the molecules to sort of line up, but in SLS, because the polar ends are much larger, they're sort of lollipop-shaped, they're going to start to curve inward, creating this structure called a micelle. But cell membranes aren't made of sodium lauryl sulfate. The amphipathic molecules that form the neuron membrane are phospholipids, the most common being phosphatidylcholine, C44H84NO8P. You thought the last one was big. They have a phosphate group on one end, similar to the sulfate group in SLS, but it also has 2 nonpolar tails. One like SLS with the little kink in it, and a straight one. This shape makes the molecules less lollipoppy, and so when they line up with each other, they don't curve in, but can naturally form a very large flat sheet with one polar side and one nonpolar side. The nonpolar side can also be attracted to the nonpolar side of another phospholipid sheet. This is the structure of the cell membrane, a double layer of phospholipids with the polar hydrophilic ends on the outside where they naturally hang out with water and ions and other polar molecules, but with a middle layer of waterproof, electrically insulated hydrophobic oils. This oily bag that contains all of the cell's parts, it's not just a soap film. Another important type of molecule called proteins float around in this two-dimensional fluid of the phospholipid layer. Think of these giant molecules as the nanomachines responsible for running almost all of your cellular biochemistry. The first important protein I want to talk about is called the sodium-potassium pump, and it is massive compared to the other molecules we've talked about so far. Phosphatidylcholine contains 138 atoms. Impressive, but the sodium-potassium pump is made of 1,250 amino acids, and each amino acid is made from, on average, 19.2 atoms, which makes this protein approximately 24,000 atoms large. Its job is to move potassium atoms from outside of the cell through the cell membrane to the inside, and also expels sodium ions from inside the cell to the outside. This results in the cell having a much higher concentration of potassium and a lower concentration of sodium than its surroundings. But another important result of this is for every 3 positively charged sodium ions it expels, it only imports 2 positively charged potassium ions. This results in the inside of the cell having a net negative charge compared to the outside. In physics, they call a state like this potential energy. All animal cells do this, but specifically in neurons, this forms the battery of electrical energy that can be used to transmit electrical signals. Why is sodium Na and potassium K? Well, we were already using S and P to mean sulfur and phosphorus, so we had to think of something else, and they're the abbreviations of the Latin names. Natrium for sodium and kalium for potassium. Were you wondering when I was finally going to stop talking about molecules and start talking about nerve cells? Well, stop wondering, because that is happening now. The electrical difference between the inside and the outside of a neuron is called the resting potential, and it is normally somewhere between negative 75 millivolts and negative 55 millivolts. All animal cells have a sodium-potassium pump, but neurons also have one called the voltage-gated sodium channel. Here's how it works. As long as the cell remains polarized, with the inside being about negative 70 millivolts compared to the outside, this protein does nothing. But if a voltage threshold is crossed, say higher than negative 55 millivolts, it will open for a millisecond. This allows sodium to start freely flooding inside the cell. These are positively charged ions, so their influx causes a significant increase in the membrane potential around the channel, up to positive 40 millivolts, before the channel slams shut. The depolarization triggers another membrane protein, the voltage-gated potassium channel, to open up too. This allows positively charged potassium ions to now start flooding out of the cell. This loss of positive ions brings the local membrane potential back down and even past the original resting potential, about negative 75 millivolts. The electrical spike caused by this voltage-gated sodium and potassium channels opening is called an action potential. It occurs locally to the voltage-gated ion channels, but if another voltage-gated sodium channel is nearby, the change in membrane potential can trigger its threshold, and cause a neighboring action potential to fire. And that one can cause one of its neighbors to fire, etc. This cascade of action potentials is how the spike propagates down the axon. Fun fact! Tetrodotoxin, the neurotoxin found in pufferfish, blocks voltage-gated sodium channels, resulting in paralysis and respiratory failure. Oh, that's not very fun. The resting membrane potential of a neuron is between negative 75 millivolts and negative 55 millivolts, and the action potential threshold is about negative 55 millivolts. So you can see it doesn't take much to trigger the voltage-gated sodium channel to initiate an action potential. In fact, neurons have a tendency to just fire off randomly. A certain amount of noise in a neuron is expected, and it is not a defect in the activity of neurons, but may be a key feature to their proper functioning. How quickly does the action potential propagate down the axon? Well, it depends on the diameter of the axon. The thicker ones propagate a little faster, but some can be as slow as 0.5 meters per second. For you people from the US, that's about 20 inches per second. If all of your nerves were this slow, it would take 4 seconds to feel if your toes were on fire. That does not seem sufficient. But there is a way to significantly increase the speed of signal transmission by up to 100 times. It's called myelination. Myelin is a fatty covering made by specialized cells that wrap around axons. In vertebrates, myelinated axon transmission can be as fast as 120 meters per second. Another fun fact, multiple sclerosis is a disorder where the immune system attacks the myelin of the central nervous system. These are not fun facts. And that is exactly how all neurons work, except for the ones that don't work that way. Whenever the basics of neurons are taught, we like to keep it simple. There's so much going on that it's really too much to address all at one time. So instead, we teach this idealized model, typical neuron. It's usually based on the motor neurons that tell your skeletal muscles to contract. They're very simple and well understood, which makes them a great place to start. But we are in danger of concluding that all neurons share the properties of this model. So far, what I've talked about applies to almost all animal neurons, but now I'm starting to get into territory where if we describe this simplified ideal neuron, it may not always be the same as all neurons, or even most neurons. Looking at all of the neurons that we know about, there are so many different types and significant variations of neurons. I don't have the time or the knowledge to go over all of them, but I at least want to point out where there is extreme variation from this typical student-level neuron model thing. Let's take a look at some exceptions in the retina. The retina is full of weird neurons. The retina is the neural tissue at the back of the eye that has photoreceptor neurons that detect light. They send that information to an intermediate layer of neurons for processing, like edge detection or movement detection and who knows what else. Finally, there is a layer of neurons called ganglion cells, whose axons form the optic nerve and send the visual information deeper into the brain for additional processing. Yes, your retina is part of your brain. Do you know which direction the light comes from? It comes from the side of the ganglion cells. The photoreceptors are actually at the very back of your eye. All vertebrate retinas are kind of built backward of how you might think they should be. I've talked a lot about action potentials propagating down axons, but that is not how it works for all neurons. The term action potential implies that the electrical spike sent down the axon is an all-or-nothing type of signal. It either spikes or it does not spike. Photoreceptor neurons in the retina don't do this. Instead, they send graded potentials. The size of the electrical spikes are different depending on the intensity of the light. In fact, neurons that generate graded potentials are typical in invertebrates. It was only recently discovered that some neurons in C. elegans, a roundworm used as a model organism of the nervous system, have a few neurons that have action potentials. The vast majority of their nervous system is composed of neurons with graded potentials, like photoreceptors. The next cell we come to in the retina is the bipolar cell. This has an axon, but it also has this long process on the other side of the cell body. That isn't an axon. This is a dendrite, an extension of the cell body to receive information, in this case from the photoreceptors. And these were part of the typical neuron illustration that I showed at the very beginning of this video. Dendrites are a very common part of most neurons, but not all of them. For example, you could see that the photoreceptor cells don't have them. Our simplified model neuron has one axon that branches into multiple terminals. This is a very common structure, but not universal. This retinal neuron is called an amacrine cell, and it only has one process. What is that thing? It's a dendrite. This cell has no axons. Also, this neuron down here is called the horizontal cell. All of these projections coming off of it, dendrites. So maybe we could say that, well, all neurons have exactly 0 or 1 axon, but a single axon can branch a lot. This is a pyramidal cell, an important neuron in your brain, commonly found in the cerebral cortex, hippocampus, and amygdala, but it doesn't look very much like our model neuron, does it? It does technically only have one axon, but that one axon branches like crazy. Oh my God, that was just one step. Don't worry, the other steps of neuron function will go a lot faster. Unfortunately, it's not because they're simpler, but because they're so much more complicated, there's a lot less to be certain about and a lot more variation between neurons. The location where neurons send and receive information is called the synapse. It is typically where the axon terminal of one neuron connects to the cell body or dendrite of another neuron. When the action potential that propagated all the way down the axon reaches its end, it can't directly send that electrical impulse into another neuron. How could it? We already know that the cell membrane is electrically insulated and waterproof. There has to be some other type of mechanism for the signal to be detected by another neuron, or in the case of motor neurons, a muscle gland or organ cell. The end of axons contain synaptic vesicles. These are little packages made out of phospholipid bilayer, very similar to the cell membrane, and they are filled with a special chemical to be used as a neurotransmitter. If our typical neuron is based on a peripheral motor neuron, then that chemical would be acetylcholine. When an action potential reaches the end of the axon, it initiates a molecular process that causes the synaptic vesicles to fuse with the cell membrane of the synaptic cleft, this tiny gap between the presynaptic and postsynaptic neurons. The specific molecular process of this is fascinating, and I could dive into it way more deeply, but I'm not going to do that right now, even though it's kind of cool, maybe another time. Now that the axon terminal has converted the electrical signal into a chemical signal, what detects that chemical signal? The cell membrane of the postsynaptic neuron will contain proteins that act as chemical receptors specific to a neurotransmitter. Because neurons can normally fire as fast as 200 times per second, the neurotransmitter has to be removed somehow to prepare for the next spike. It's commonly broken down by enzymatic proteins, or the axon terminal can actually suck it back up into new synaptic vesicles. This neurotransmitter recycling is also beneficial because neurotransmitters are only manufactured in the cell body, and it is not easy to transport them all the way down these axons to their terminals. Neurotransmitter uptake can save the neuron a lot of time and energy. And there are a lot of other types of neurotransmitters, but there isn't even just one type of acetylcholine receptor. At the synapse between a motor neuron and a skeletal muscle cell, also known as the neuromuscular junction, acetylcholine is fast-acting and excitatory. It causes ion channels to depolarize the cell, increasing the likelihood of an action potential in the postsynaptic cell. But there is a different acetylcholine receptor protein found in cardiac muscle and a few other places that is slow-acting and has an inhibitory effect on the chance of an action potential. And that is only one of over 60 identified neurotransmitters in the human nervous system. These include biogenic amines like dopamine, epinephrine, serotonin, and histamine, amino acids including GABA, glycine, glutamate, aspartate, neuropeptides like insulin and opioids, and nitric oxide. Sad fact, Curare, a plant-derived arrow poison from South America, blocks acetylcholine receptors at the skeletal neuromuscular junction. Sufficient poisoning results in paralysis and death by suffocation. Our model neuron only uses one type of neurotransmitter, but that doesn't mean all neurons have to. According to Neuroscience, 2nd edition, until relatively recently, it was believed that a given neuron produced only a single type of neurotransmitter. There is now convincing evidence, however, that many types of neurons contain and release 2 or more different neurotransmitters. There are now numerous examples of different peptides being present in the same terminal, as well as cases in which 2 small molecule neurotransmitters are found within the same presynaptic ending. As if 60 neurotransmitters aren't complicated enough, now they can come in cocktails of multiple different ones that don't even need to be released simultaneously. For example, a low frequency of spikes could release some synaptic vesicles, while higher frequency spikes could trigger all of them. Our typical neuron with synapses at the axon terminals connecting to dendrites of another is quite typical, but not exclusive. Dendrite to dendrite, axon to axon, and even dendrite to axon synapses are possible. Axoaxonic synapses seem to be important to many areas of the nervous system, allowing one axon to moderate the synapse of another. In skeletal muscle, acetylcholine is an excitatory neurotransmitter. If enough acetylcholine receptors are bound, it will reach a threshold and initiate an action potential. Other neurotransmitters and their respective receptors are inhibitory. They can reduce the chance of initiating an action potential. Neurons that have both excitatory and inhibitory receptors in their synapses can be thought of as doing simple addition and subtraction, and then initiate the action potential if the difference between the excitatory and inhibitory signals are greater than some threshold. In fact, this is what inspired the McCullough-Pitts artificial neurons used in most artificial neural networks today. But we now can see that natural neurons are much more complicated. With over 60 different types of neurotransmitters, this seems excessive if all we need is simple addition and subtraction. And we now know that each neuron can perform complex mathematical calculations. The abstract of the paper Neuronal Arithmetic states, yes, I'm reading the whole thing. It's very good. You'll like it. The vast computational power of the brain has traditionally been viewed as arising from the complex connectivity of neural networks, in which an individual neuron acts as a simple linear summation and thresholding device. However, recent studies show that individual neurons utilize a wealth of nonlinear mechanisms to transform synaptic input into output firing. These mechanisms can arise from synaptic plasticity, synaptic noise, and somatic and dendritic conductances. This toolkit of nonlinear mechanisms confers considerable computational power on both morphologically simple and more complex neurons, enabling them to perform a range of arithmetic operations on signals encoded in a variety of different ways. As alluded to in that abstract, neurons can also make persistent changes to themselves, altering how future calculations may be performed and how information is sent to other neurons. Many neurons do not perform the exact same function for their entire existence. Their function can change based on what has happened to them in the past. Propagation of action potentials and synaptic transmission seem to be very well documented, all the way down to the molecular level. I could have gone much deeper on their specific mechanisms, but when you try to find out more about how postsynaptic receptors and the neural cell body perform calculations, it gets a lot more complicated. What even the hell is this? It's easier to think of neurons simplistically, with complex computation arising from the connections between them, thinking like the neuron is the transistor of the brain. But we simply know that isn't true. We know of the existence of single-celled creatures with simple but intelligent behavior. Why would a complex molecular machine just be reduced to a transistor of the brain? I think a better analogy of neurons to electronics is that of the integrated circuit. That is a component that can accept multiple inputs, perform simple calculations, and send the results to multiple outputs. The brain has 86 billion of these. At that size, that would be enough to fill about 24 Olympic swimming pools. So, it might be a while before we can replicate the human brain in a computer. Thanks for watching to the end. I really appreciate it. If you want to learn a little more about neuroscience, here's another video I made. Until next time, please care for yourself and make good decisions.